Hi, my name is Phoebe Lostro, and I'm a biology professor at Colorado College. And this brief screencast is intended to introduce you to the Kyoto Encyclopedia of Genes and Genomes. To begin, go to www.genome.jp/KEGG/KEGG2.html, and that will bring up this home screen. The KEGG database contains millions of gene and protein sequences, and the purpose of the database is to make it possible for you to compare a gene or protein that you care about with this entire set in order to make functional and evolutionary hypotheses about the gene or protein that interests you. The best way to start is with a gene that you know you're interested in. And so for this purpose, I'm going to type in the name of a gene that I often study called ACIAD 0167. Click Go, and here's the page for 0167. And I'm going to click on the link here. Okay. This is the main page about gene ACIAD 0167. You can see that it's in the organism Acinetobacter species ADP1. This is an organism that I study in my microbiology research lab. And then here's a list of a lot of things that we know about this protein. So first of all, you can see in the column here the amino acid sequence for this protein. So it starts with methionine, like all proteins do, and it ends with a lysine. So you're looking at one-letter amino acid codes for this protein, which is 1,106 amino acids long. You can also find the nucleotide sequence that encodes this protein in the window below the amino acid sequence. It's called NT sequence, or nucleotide sequence. Furthermore, if you want to know the nucleotide sequence surrounding the gene of interest, you can use these boxes and type in however many nucleotides you want. So for this example, I'm just going to show you 100 nucleotides upstream of the gene. So now when I click on nucleotide sequence, the first 100 are upstream of the gene number 167. And then gene number 167 sequence starts here. Oops, didn't mean to pick that up. Starts here with this ATG. Okay. Just click the back arrow to go back. And I'm going to scroll up to show you a few more things up near the top. Okay, so the first thing you should notice are these buttons that say ortholog, paralog, and motif. Orthologs, I'm going to click, are defined very specifically in the KEGG database as proteins that have amino acid sequences that are significantly similar to your protein and are found in a different organism. So when I hit ortholog, it did a huge search against every other protein in the database, and it found all of these proteins that are statistically similar to gene number 167. And look, you can scroll down. There's hundreds of these. Okay? And these are all statistically similar enough so that for the ones that share identity over a large region of the protein, they probably fold into a very similar structure and have a very similar function. So an important thing to notice is len, L-E-N. That stands for length. So remember, our gene was 1,100 something amino acids long. And notice that for all these hits up top here, you're looking at between 900 and 1,000 amino acids that are also similar in these proteins that are listed over here on the left. So in other words, these proteins probably are quite similar to one another. So you can actually look at the alignment for how all of these proteins align with 167 by first selecting the proteins you want to look at, and I'll just select the top 5. Incidentally, these are arranged in order of the most similar protein in the database down to the least similar, yet statistically significantly similar protein in the database. So let's just look at the top 5, and I'm going to choose ClustalW, which is an acronym. It's just a very common computer program for aligning amino acid sequences to see how they're similar. So I just hit select and the computer thinks for a while. And here it has drawn the alignment for me. So first I can scroll down and just look at the alignment in black and white. So my gene, remember, was ACI-AD0167, which is down here. And then here are those 5 proteins that it's similar to. AB57_3817, ABAYE0118, and so on. So each of these proteins has its own name, and each of them is in a different genome. And then when you look at the alignment here, what you see are lots of stars, periods, and colons. So the stars are positions at which our protein is identical in sequence to the other proteins. Colons are positions at which the side chain of the amino acids has similar chemistry. And so the F and the L's in this column are considered to have similar chemistry because they're both hydrophobic. And then the period is saying that the side chains on these amino acids too are chemically similar in some other way that is a little bit less similar than the colon. And so the stars mean identical, the colons mean very similar, and the periods mean a little bit similar at that position. And so when you look at all these proteins, you can tell that there are really some regions of these proteins that are absolutely conserved. Okay? And then there are some other places where there's a lot less conservation, which means you see spaces in the alignment where there's an A or a D here, an A or an N, an L or an A T or an M, and so on. But actually these proteins are so similar that it's hard for me to find a region of the protein that is less similar to these other proteins. So it's a pretty strong alignment, strongly suggesting that these proteins are evolutionarily descended from the same common ancestor and that they are very likely to have a similar function. Okay, so there is another way to represent this information visually, which is to draw an evolutionary tree in which the pattern of branching represents the similarity of each sequence to every other in the alignment. And what you do is scroll up to the top, and here's the Select Tree menu. And I'm just going to use the rooted phylogenetic tree, but you could choose one of these others. This would be called neighborhood joining, and I can't remember what UPGMA stands for. It's an acronym. But before you use one of these trees in any kind of presentation or written report, you should probably look up these kinds of trees in a textbook on evolutionary biology or online and find out what they are so that you can use them properly. But for now, let me just show you the rooted phylogenetic tree. And what you find out is that these two sequences are more similar to one another than they are to this third one. And these 3 are more similar to each other than they are to these 2, and so on. All right, and up here you can output those files to be able to use them in a presentation or a written report. Of course, you would cite the KEGG database and probably also ClustalW in order to provide a citation for this work. I'm just going to close out of this window and out of this one and get us back where we started so that I can take a pause and say I'm going to stop for now, but I will record other screencasts to show you what happens when you use the paralog button, the motif button, the genome map button, and the gene cluster button. But for now, I'll just say thanks for your attention, and I hope that you have enjoyed this introduction to the KEGG database.